Welcome to your first semester of zoology. Today, we're diving into the fascinating world of helminths, commonly known as flatworms. Imagine trying to survive without lungs to breathe or a heart to pump blood. It sounds impossible, right? Yet these incredible organisms have mastered survival through absolute simplicity. By stripping away complex internal systems, they have adapted to thrive in damp environments and even inside other animals as parasites. Let us explore how their unique body plan makes this possible. Now that we know what helminths are generally, let us look at their specific biological classification. Helminths belong to the phylum Platyhelminthes. One of their most important evolutionary advancements is that they are bilaterally symmetrical. This means if you draw a line down the middle of their body, the left and right sides are mirror images of each other. Why does this matter? Before this evolutionary step, organisms like jellyfish were radially symmetrical, meaning they interacted with their environment from all sides equally. Bilateral symmetry allows these flatworms to have a distinct head and tail, enabling directed, forward movement to actively hunt for food or find a host. Building on their symmetry, let us examine how their bodies are constructed from the inside out. Helminths are triploblastic, which means their entire body develops from exactly 3 primary embryonic cell layers: the outside ectoderm, the middle mesoderm, and the inner endoderm. However, despite having these 3 layers, they are strictly acoelomate. In many animals, including humans, there is a fluid-filled cavity between the gut and the outer body wall that holds organs. Helminths completely lack this cavity. Instead, that internal space is packed completely solid with a spongy, supportive mass of cells called parenchyma. This solid construction is a defining hallmark of their anatomy. Understanding this solid body plan is not just academic, it is absolutely critical for global medicine. Why should we care about these worms? Because many of them are devastating parasites. Organisms like tapeworms and liver flukes cause severe, sometimes fatal diseases in humans, pets, and livestock. For instance, a disease called schistosomiasis is caused by a blood fluke and affects hundreds of millions of people, causing chronic illness and economic devastation in developing nations. By studying their unique physical traits, scientists can develop targeted drugs. On the flip side, free-living flatworms like planaria possess a seemingly magical ability to regenerate entire body parts from tiny fragments, offering vital clues for human stem cell research. This brings us to a fascinating biological puzzle. Since helminths lack a circulatory system to pump blood and a respiratory system to breathe, how do their internal cells get oxygen? The answer is passive diffusion. Oxygen must seep directly from the outside environment, across their skin, and travel to every single internal cell. But there is a catch. The efficiency of this passive diffusion depends entirely on the shape of the animal. If a worm is too thick, the oxygen gets used up by the outer cells before it can ever reach the deep inner core, causing those inner cells to suffocate. This physical limitation dictates their entire body shape. To truly understand why they must be flat, we have to look at the mathematics of biological diffusion. This relationship is defined by a simple but powerful formula. R equals A divided by V. Here, R represents the surface area to volume ratio. A stands for the total surface area of the organism's body, which is essentially how much skin is available to absorb oxygen. V represents the total internal volume, which is the amount of tissue that needs that oxygen. In plain English, this formula tells us how much outer skin is available to supply vital oxygen to the internal tissues. A larger value for R means oxygen can easily and quickly reach all internal cells without needing a pumping heart. Let us see this formula in action with a concrete example. Imagine a hypothetical worm that is perfectly cubical with a side length of 2 millimeters. We want to find its surface area to volume ratio. First, we calculate the total surface area. Since a cube has 6 identical square faces, we square the side length of 2 to get 4 and multiply by 6. This gives a total surface area of 24 square millimeters. The internal volume of this cube is 2 times 2 times 2, which is 8 cubic millimeters. Dividing the area of 24 by the volume of 8 gives us a final ratio of 3. This means there are exactly 3 square millimeters of skin available to serve every single cubic millimeter of internal tissue. Now, let us compare that thick worm to a flattened helminth. This flatworm has the exact same internal volume of 8 cubic millimeters, but its dimensions are stretched out to 8 millimeters long, 1 millimeter wide, and 1 millimeter high. We calculate its surface area by adding the areas of all 6 rectangular faces. We have the top and bottom, plus the 4 thin sides. Adding these together gives us a total surface area of 34 square millimeters. Notice how the area has expanded just by changing the shape. We then divide this new area of 34 by the constant volume of 8. This yields a final ratio of 4.25. Physically, this means the flatworm has significantly more skin available per unit of internal volume. So, we have calculated the ratios, but what does this actually prove biologically? By comparing the two results, we see that 4.25 is significantly higher than 3. This mathematically proves that by flattening its body dorsoventrally, the helminth successfully maximizes its surface area relative to its volume. This is not just a random physical trait, it is a strict mathematical necessity. That relatively low number for the cubical worm indicates its inner core is too far from the oxygen-rich outside environment, and it would quickly perish. The flatworm's high ratio allows sufficient oxygen to diffuse directly through the skin to all internal cells, perfectly explaining the evolutionary necessity of the flat body plan in Platyhelminthes. Now that we understand their shape, let us look at how they survive as a species. Parasitic helminths, such as tapeworms, face a massive biological challenge. The journey from one host to another is incredibly dangerous. The chances of their eggs successfully reaching and infecting a new host are microscopic. Most eggs will dry out in the sun, be eaten by the wrong animal, or be destroyed by a host's immune system. Because the environment is so hostile, these parasites cannot rely on just a few offspring. They must exhibit extreme reproductive output, a concept known as high fecundity, to ensure that at least a few survive to continue the species. To grasp the sheer scale of this challenge, we can use a basic probability expectation formula from population biology. The formula is S equals E times P. Here, S represents the final number of successfully established offspring. E represents the total number of eggs produced by the adult parasite in a given time frame, which is their biological investment. Finally, P represents the mathematical probability of survival for each individual egg. By multiplying the total attempts, which is the number of eggs, by the success rate of a single attempt, we find the expected value. If the probability P is extremely low, the egg count E must be massively high, just to keep S above 0. Let us apply this formula to a real-world scenario. Suppose a single adult tapeworm produces a massive biological investment of 50,000 eggs every single day. However, due to extreme environmental barriers, the probability of a single egg surviving is a microscopic 0.00004. We want to find the expected number of successful offspring. We substitute our known values into the equation, multiplying 50,000 by 0.00004. This simple multiplication yields exactly 2 successful offspring per day. Despite the massive initial number of eggs, the harsh reality of the parasitic lifestyle reduces the final success rate to almost nothing. This perfectly demonstrates why their reproductive organs take up almost their entire internal body space. To tie all these concepts together, let us look at a cross-section of a helminth. This diagram perfectly illustrates the acoelomate body plan we discussed earlier. Notice the flattened oval shape, which we proved is necessary for oxygen diffusion. On the very outside, you see the thin ectoderm, or epidermis. Right in the center is the endoderm, which forms the gut. But the most important feature is what lies between them. Look at the thick region, labeled mesoderm, or parenchyma. Notice how it is completely solid and packed with spongy tissue. There is absolutely no empty, fluid-filled cavity here. This solid packing, combined with the flattened shape, defines the unique physical reality of being a flatworm. We have covered a lot of ground today, so let us summarize the key takeaways. First, helminthes are uniquely defined by their bilaterally symmetrical, triploblastic, and acoelomate body plans. Second, we learned that their signature flattened shape is not an accident. It is a strict mathematical necessity to maximize surface area for diffusion, since they lack a heart and lungs. Third, their internal space is packed completely solid with parenchyma tissue. Finally, we saw mathematically why parasitic forms must exhibit extreme reproductive output to overcome the incredibly low odds of transmission. Understanding these general characters provides the essential foundation you need to study their complex life cycles and their profound impact on global health. Great job today.